Hello everyone. Today's topic of discussion is Riccia thallus. Let us see what are the contents we are going to deal today. We will start with the habitat of thallus and we will see the external and internal structure of thallus. Then we will study about the reproduction of thallus followed by the alternation of generation. Now let's see the classification of Riccia thallus. It belongs to the kingdom Plantae. So the group have the presence of chlorophyll. It belongs to the subkingdom Cryptogams. That means they belongs to the non-flowering plant group. It belongs to the division Bryophyta. That is the lack of true root and true vascular tissue. Class Hepatocopsida. That means the thallus is gametophytic. Dorsiventrally flattened structure. Order Marcantiales. That is, the characteristic feature of Marcantiale is that they must be dichotomously branched and the scales must be present. Family Ricciaceae, which means they must show air pore that should be simple. Sex organs must be seen in the mid-dorsal groove, and finally the sporophyte will be in the form of capsule. Coming to the habitat of Riccia. The genus Riccia comprises about 138 species which are cosmopolitan and widely distributed both in tropical and temperate regions of the earth. With the exception of single aquatic species Riccia fluitans, all the other species are terrestrial in nature. About 28 species of Riccia have been reported from different parts of the India. Of this, the common species are Riccia discolor, Riccia crystalline, Riccia panthocontensis, Riccia melanospora, Riccia forti and Riccia gangetica. Out of this, Riccia crystallina and Riccia cassiopi, Riccia pandal are endemic in India. Riccia mainly grows on damp soil and shady places and other similar terrestrial habitat. Let's see the external structure of thallus. The plant body of Riccia is flat dorsiventral thallus, which is prostrate, ribbon-like and more or less fleshy in nature. The branching of the thallus is dichotomous. Due to the repeated dichotomy and also due to the occurrence of the number of dichotomously branched plants together at the place, the plant often takes a rosette form. The thallus is usually green in color and each thallus is provided with a midrib in the middle portion, which forms the apical notch at the tip. Now let's see the external and internal structure of rhizoid in Riccia thallus. The ventral, that is the lower surface of the thallus, bears numerous multicellular hairs and scales. This unicellular rhizoid is of two types. One is smooth-walled rhizoid and another one is tuberculated rhizoids. This rhizoid attaches the thallus to the substratum and absorbs water and solute from the soil. The scales in young plant are arranged in one transverse row near the apex. When the thallus matures, they form two rows near the margin of the thallus. Now let's start with the internal structure of Riccia thallus. The vertical section of the thallus shows an outline with a depression in the center, termed as the dorsal groove. Externally, simple thallus shows a slightly internal differentiation. The thallus is formed of two different regions. They have a distinct region. The lower one is, that is the ventral, is storage region, and the upper or dorsal is photosynthetic. It is also called as assimilatory region. Storage tissue, which is the lower or the ventral region in cross section, shows colorless, compactly arranged parenchymatous tissue. It is without any intercellular spaces. The cell of the parenchyma tissues generally lack chlorophyll and they contain starch. Therefore, the ventral region primarily constitutes the storage tissue. The lower portion of the storage region is bounded by a single layer epidermis from which rhizoids of two types, smooth walled and tuberculated, arises. The lower epidermal cells also have ampigastria. The function of ampigastria is to give protection to the surrounding tissues. Now we will see the primary functional area of Riccia thallus, that is photosynthetic region. It is present on the dorsal surface of the thallus. It consists of chlorenchymatous cell arranged in vertical row of one cell thickness. This row of chlorenchymatous cells are called as photosynthetic filaments. It is between the chlorenchymatous row there are air chambers, air canals and air clefts. The air chamber lacks photosynthetic filament. The air cleft, it is surrounded by 6 to 8 vertical row of chlorenchymatous cells. The outermost on terminating cells of each row is slightly bigger and colorless in nature. These terminal cells together form an interrupted upper epidermis. The air cleft communicate with the outer environment through the gap in the upper epidermis called air spores. In case of aquatic species, the epidermis is continuous and the air pores are absent. So, the air pores are present in the terrestrial habitat of Riccia. The thallus shows large air cavities which stores air and gives buoyancy to the thallus. The epidermal cells of the photosynthetic region also shows the presence of chloroplast. Now, let's learn about the reproduction in Riccia. The Riccia reproduces by vegetative and sexual method. Vegetative reproduction is by fragmentation, adventitious branches, tuber and persistent apices. Now first we will see the vegetative reproduction by fragmentation. This kind of reproduction is depending on the aging of vegetative cells. The cells in the older portion dies, that is, it becoming old age, and eventually started disorganizing themselves. When the death and decay of the older portion of the thallus reaches the dichotomy, the end lobe becomes separated. Each of this by apical growth grow into a new thallus. Now, the most common type of vegetative reproduction is adventitious branching. In Riccia fluitans, special adventitious branches arises from the ventral surface of the thallus. It arises in the midrib region. They become detached from the parent thallus by the decay of connecting tissue and form a new plant. Now, let's see the third type of vegetative reproduction in Riccia thallus, that is tuber formation. It has been reported in Riccia perennis, Riccia discolor and Riccia vescata. The apexes of the thallus lobe become thickened to form tubers at the end of the growing season. With the advent of unfavorable condition, the plant dies. The tumor, which is thick-walled in nature, remain dormant and resume growing under suitable condition and develop into a new thallus. This is followed by the persistent apices. Prolonged dry condition will lead the thallus die, except the apical part. This apical part, however, grow deeper into the soil and they become thick. At last, the active part, when the favorable condition turns, will develop into a new thallus. Now, let's learn about the sexual reproduction in Riccia. Riccia is mainly homothallic, that is monoecious in nature. That is, same plant body bears both male and female reproductive organ. The male reproductive organ is called antheridia and they produce anthrozoid. The female reproductive organ is archegonia and they produce the gamete ovum. Some species like Riccia discolor, Riccia fructi are heterothallic in nature, that is they are dioecious, or it is also called as unisexual. In monoecious species, both archegonia and antheridia arises singly and each lie in the floor of the separate cavity. This sex organs are in dorsal surface. They are deeply sunk in the mid furrow of the thallus. Antheridium, it is the male reproductive part. Each antheridium stands in a deep pit called antheridial chamber and it is attached to the thallus tissue by means of multicellular stalk. This antheridial chamber opens at the tip by a narrow pore called ostiole. The antheridium is an ovoid or pear-shaped body enclosed by a single layer of sterile jacketed layer known as antheridial wall. Inside the jacket layer lies the numerous anthrozoid mother cells, which later metamorphoses into two sperm or spermatid or anthrozoids. The anthrozoids or the spermatozoids formed in the antheridium is a minute, slender, curved and flagellate structure. Flagella are 2 in number and are inserted in the anterior region of the anthrozoid. The body possesses elongated blebboblast and nucleus and a little cytoplasm. The unused cytoplasm remain attached to the posterior end forming a vesicle. Now let's see the female reproductive organ of Riccia, that is archegonium. Each archegonium also stands in a pit called archegonial chamber. The archegonium is without stalk and directly attached with the thallus tissue, that is in between the photosynthetic region. The mature archegonium is having a short stalk, flag shape and consists of venter and neck. As we studied a few minutes back, archegonium is formed of 3 parts: stalk, venter and neck. We already know the stalk is small and few cells, but the venter is broad and the venter wall is 1 cell in thickness. The venter wall encloses a cavity called venter cavity, which consists of 2 unequal cells. The first cell is very smaller and we call them as a venter canal cell, and the larger one is posterior ovum. It is also called as egg cell. Then neck consists of 6 vertical row of cells. At the tip, 4 specialized cells, that is lid cell or the cover cells, are present. After the gamete formation by the male and the female reproductive organ of the Riccia, the dehiscence of the sex organ should take place. For that, water or even moisture is very necessary. Spermatozoid, which lie in the antheridial cavity, because the wall of the spermatid which has been already dissolved. Water finds its way to the antheridial cavity through the ostiole. Now the antheridia burst, liberating anthrozoid due to the pressure created by the antheridial wall. The archegonium, which matures at the same stage, will start developing now. That is, the ventral canal cell and the neck canal cell disintegrate or degenerate to form mucilage. The mucilage absorbs more water and pressure is created now. This will help in separating the lid cell. Now the sperm reaches the egg cell. Certain chemical substances are exuded along with the mucilage, which leads to the separation of the lid cell. That helps the sperm to reach the egg cell for an easy fertilization. The chemical substance which was exudated along with the mucilage was probably the chemical that attracted the spermatozoid. Only one of the anthrozoids surrounding the egg succeeded in uniting with the egg. Thus, the gametophytic stage ends with the process of fertilization. Later, the zygote is formed. The zygote is the first cell of the sporophytic generation. It is embedded in the venter. With the formation of a zygote, diploid sporophytic generation begins. Zygote, it is the first cell of the sporophyte. Immediately after the formation, the zygote enlarges in size until it nearly fills the cavity of the venter. The mature sporophyte of the Riccia is the simplest among the sporophyte. It consists of only capsules or the spores, while the food and the setae are the absent. Here, the sporophyte is embedded in the storage tissue of the gametophyte and enclosed in the ventral cavity of the archegonium. Now, the zygote nucleus first divides transversely into two cells, each which again vertically divide to form 4-cell structure, that is quadrant stage structure is formed. Then another vertical division takes place, so 8-cell structure, that is octant, is formed. The cells of the octant divide and redivide irregularly, forming spherical mass of 20 to 410 cells. The periclinal division takes place, which cuts off the outer layer of the cells called amphithecium and the inner round mass of cells called endothecium. This amphithecium first constituted the jacket of the capsule, but later it serves as a nutrition of the spores. The outermost layer constituted the jacket of mature sporophyte. The endothecium develops into sporogenous tissue. The sporogenous tissue later develop into mother cells, that is spore mother cells. From each spore mother cells, two successive divisions will take place, one of which is reduction division that results in 4 haploid spores. Some of the few cells of the sporogenous tissue remain sterile and form a nurse cell. The mature sporophyte is a globose capsule-like structure without any fruit and setae. It is a simple spore case, that is, the spore case is a capsule provided with a sterile jacket layer of 1 cell thickness. Inside the jacket layer lies the spore tetrads and the nurse cell. The spores are liberated after breaking the surrounding tissue of the thallus during the favorable condition. After liberation, the spores will start germinating during the favorable condition. After the decay of the calyptra, the spore remains in the soil. Once the favorable condition turns, the spore starts germinating. The spores are tetrahedral in shape. The spore wall, that is sporoderm, is formed of 2 layers. The outer wall is called as exine. It is also called as exosporium. And the inner wall is called as intine. It is also called as endosporium. Exine is variously ornamented. Exine is cuticularized with a thick wall, cutin, while the intine is thin-walled. On the turn of the favorable condition, the spore starts germinating. For that, they need light and presence of moisture. Now, the spore starts absorbing water and the pressure is created by the intine, which will make the exine wall rupture. This intine comes out for forming a small germ tube. The content of the spore now started migrating to the tip of the germ tube, and the partition is now made. This will make the cells to divide and redivide at the apical region that will result in formation of new gamete, resulting in gametophytic generation. we have now come into the last slide, that is alternation of generation in Riccia. In Riccia, there is two distinct generations in the life cycle. They are haploid, that is gametophytic generation, and diploid generation, that is sporophytic generation. Gametophyte, that is antheridia and the archegonia, develop anthrozoid and ova respectively. This anthrozoid unite with the egg, resulting in the formation of zygote. The zygote is diploid. Sporophyte, a diploid phase, is a complete parasite depending on the nourishment in the gametophytic tissue. Sporophyte, a capsule. Within it, after reduction division, gives rise to a number of haploid spores. Now, this haploid spores germinate in the favorable condition to develop into a gametophyte. Gametophyte give rise to sporophyte, and the sporophyte give rise to gametophyte, and like this the alternation will take place with each other. Thank you. In the next video, we will learn the structure and reproduction of anthocerox.
Want to know more about these topics? Please follow us on Twitter, Facebook, Instagram, and don't forget to subscribe our channel.